morning. I'd like to welcome everybody to Medicine Grand Rounds for our special lecture in cardiology, the Guller Family Cardiovascular Disease Prevention Lecture. And before I introduce our speaker, I do want to just say a few words about this endowed lecture. The Gullers have been a fixture in St. Louis. They founded a homegrown manufacturing company called Essex Industries, and that's been passed on through now 3 generations. But as many folks in the country, both Cindy and Keith had their brush with either heart disease or cardiovascular risk factors. And with that experience, they really wanted to establish this lecture to provide and promulgate the latest evidence on prevention of cardiovascular disease. And so this lecture was established. And I'd like to recognize Cindy and Keith, who are in the audience, as they have been for every as they have been for every lecture, including this 13th annual Guller lecture. With that, it's really my pleasure to introduce our speaker, Dr. Pradeep Natarajan, who is Director of Preventive Cardiology and the Fireman Endowed Chair in Vascular Medicine at Mass General, Associate Director of Personalized Medicine at Mass General Brigham, Associate Professor of Medicine at Harvard, and Associate Professor of Epidemiology at the Chan School of Public Health, and Associate Member of the Broad Institute of Harvard and MIT. His training, he received his undergraduate at UC Berkeley, his MD at UC California, and his MMSc in biomedical informatics at Harvard. And he completed his clinical training and his research training and fellowship in cardiovascular medicine, internal medicine at Brigham and Women's, and cardiovascular medicine at Mass General. I think it's fair to say that Pradeep has expanded the scope of how we think about prevention and has really moved the field to examine new risk factors with new perspectives. And he does this using essentially genomics, but a large variety of techniques, including genetic epidemiology, large-scale sequencing studies with genotype-driven human investigation, and genetic testing implementation. He's very well versed in both monogenic and polygenic characterization of heritability, including polygenic risk scores through whole genome sequencing. And he's also more recently moved into the field of somatic mutations, hematopoietic stem cell mutations, contributing to cardiovascular disease. He's an associate editor for JAMA Cardiology and Science Advances, and he's authored over 350 scientific papers in leading journals and has been very well funded through the NIH and other organizations. He's a member of the American Society for Clinical Investigation and has received multiple awards, including the Joe Vita Award from the American Heart Association and 2 mentoring awards from Harvard. It's really my pleasure to invite Pradeep to come and deliver the Guller Lecture this year. His talk's entitled Genetics to Advance Cardiovascular Medicine.
Thanks. All right, thank you so much for the invitation. I was saying the last time I was here, I was interviewing for medical school. I don't remember if I got in, but there's probably a 50-50 chance. But I am super excited to be here. I interface with a number of folks here, so really nice to see people in person. These are my disclosures, more readable, but not relevant to this talk. So the title of this talk, Genetics to Advance Cardiovascular Medicine, is really kind of a synthesis of a book chapter I wrote for Dr. Braunwald's textbook, Braunwald's Heart Disease. And there'll be a new version, I think, this year. And I won't be able to go through every single method or approach that we use human genetics, but human genetics we use as a tool to understand cardiovascular disease because it is a very robust tool for causal inference, just short of randomized controlled trials, the gold standard for causal inference in humans. Natural occurring human genetic variations established at conception and basically fixed across the lifetime. So normally when you look at an exposure and an outcome, there may be many different reasons that explain a relationship between the two. Maybe a correlation that reflects other important features that are true causal relationships or itself may be directly causal. With human genetic variation, if there's an association with the phenotype of interest, that is fixed features, is nothing else that is going to change the allocation of those alleles, and that can be a robust tool for causal inference. Now, this is the mall close to where I live in LA, fortunately a good distance from where the wildfires are, but actually about 10, 15 miles. And I show this just as kind of a point for why I'm doing the things that I'm doing, because people ask me all the time, why am I a cardiologist? Why am I a preventive cardiologist? Most people have not heard of a preventive cardiologist. I certainly did not really until I was a clinical fellow. But I was motivated pretty early on about coronary artery disease, just because it was very common among the people that I saw in my community, in the South Asian community and also beyond. And so I was really interested in understanding coronary artery disease, but really helping my community. Now for bonus points, this is actually a mall that probably everybody has seen. Does anybody recognize this mall, this parking lot? Awesome, that's exactly right. That's where Marty McFly first went back to the future. So back then, actually not that many people were going to the mall. Still not the most popular mall, but has been the set for a few TV shows and movies, like many things in Los Angeles. But this is kind of our claim to fame. The other claim to fame was, this is also where the Black Eyed Peas, if anyone remembers the Black Eyed Peas, got their start. Fergie went to my high school, was a couple years older than me. Has achieved a different level of excellence. So, I never really understood, for a long time actually, how science or understanding coronary artery disease could merge together in clinical medicine. Both of my parents are PhD researchers. They came to the US, they came to LA, USC for their postdocs. My mom's a basic scientist, chair of her department, and my dad left the sciences because the funding was not great. So they very much discouraged me from doing science, and they said, instead become a doctor, because the people in our community who were doing well were physicians. I think what changed for me is when I was in college and really developed a real love for biochemistry. And the people who were teaching the courses were Jennifer Doudna, Carolyn Bertozzi, Randy Sheckman. These are all people who ultimately became Nobel laureates. But were also amazing teachers. And so I had great opportunities to start to understand how the things that I was learning in class and to trying to apply to models of disease. And at that time, what I was focused on, not because I had some grand plan, was really focusing on exceptional Mendelian families, families that had interesting lipid patterns, who had also interesting segregation of coronary artery disease, to try to understand what was driving those patterns and would those have broader insights. And the tools that I use today in human genetics, this was not something that anyone could use at that time. But these were really foundational experiences for me to suggest that there could be a connection. But I was very much fixated on kind of making it to the end. I don't think anyone really told me how long it was going to be. And I was still very much motivated to become a good clinical doctor. Many, many years later, basically going through the California public school system from kindergarten to medical school, have now landed in Boston, been there for about 17 years. And I show this map because, you heard from my intro, lots of different institutions, and so it's not always clear how things organize, but I know many of you folks have been in this area. So here is, here's obviously a picture of Boston here. This is where MGH is. This is where I spend most of my time. This is where I have my clinic. Part of my lab is within cardiology in the Center for Genomic Medicine, and then split with the Broad Institute, which is an interesting freestanding human genetics research institute that has served as a node for lots of different human genetics, population genetics and human genetics like I do, but also assay development and genomic editing technologies have all been central here, at least in Boston, and it draws people from all over. Because of the proximity, there has been historically a stronger connection, at least with us at MGH. The Brigham is here and still have lots of collaborators. I was a resident there when Nate and Karen were fellows at the Brigham. And I do teach in human genetics at Harvard Medical School. And the things that we're teaching is obviously far different than any of us learned in medical school. But I do spend a lot of time with the medical students and the undergraduates because those very early foundational experiences substantially influence what I was doing later on, and if they don't get those experiences earlier, then we're going to lose them to other fields or other things that will then excite them. And then more recently, spend my time at this entity here. This is also part of our health system. This is the Mass General Brigham Personalized Medicine. It's the entity that oversees our biobank. Our biobank's now about 160,000 individuals, 60,000 with human genetic information connected to our EHR, consented for callback, and is among the largest healthcare-associated biobanks in the US. And now I'm the associate director for that, and then all the in-house genetic testing, and some of that will become relevant later in the talk. So there's a lot of triangulation, and then an equal amount of time dropping my kids off in school, which is somewhere over here. So as I said, I was excited about kind of the basic science and connecting biochemistry to exceptional families, but really motivated also because there was nobody in my community, nobody around me who kind of did both together. And I was really excited and really passionate about becoming a good clinical doctor. But one thing that I increasingly observed, and this is not earth-shattering, is that there's lots of equipoise in medicine, tons of outstanding questions, and I had felt that as I was seeing more, immersed in academic medical centers, that there were people that were taking those observations, taking those questions, and starting to address them in different settings. It was really powerful to me, and that directly connected back in improving the care for those patients. So, some of the early observations that were foundational for the work for me was really rooted in observations of family history being enriched for patients who had premature coronary artery disease. There are lots of different types of cardiovascular diseases that people were suffering from in the early 20th century, often complications of rheumatic heart disease, infectious endocarditis, hypertensive heart disease, syphilis, but really starting to take off in the early 20th century into the 1950s were complications of coronary artery disease and myocardial infarction. Sufficiently so that people could start to write case reports of early onset myocardial infarction, and this is among the first case control studies. This is one, there's a kind of description from Samuel Levine from the Peter Bent Brigham Hospital, and then another, a collaboration with him and Paul Dudley White, the founder of our unit at MGH, with the cardiology fellow, taking all these cases from the Boston area hospitals that were coming in. And a recurrent observation is that those who were suffering from premature events also often had a family member who also had a premature event. Now, this is a great article for, if anyone is interested in cardiovascular disease, is Paul Dudley White's perspective in his heyday of where cardiology has moved up until then, from 1628. And what is remarkable is kind of where he ends the article, really calling out family history, and most specifically, he uses the word human genetics. We need to think about the host. We're thinking a lot about the extrinsic factors, which are obviously important, but we have not we've not spent a lot of time thinking through the innate features that are driving cardiovascular disease in our patients. But it is, pretty remarkable that he called out human genetics, because human genetics as it stands today, at that time, was very much in its infancy. We are in a world that is really kind of set by Gregor Mendel, an Austrian monk, but not too long before Mendel, and really during his time, we were in an earlier phase by Jean-Baptiste Lamarck, or Lamarckian genetics, which is kind of this notion that if a parent experienced something, the giraffe stretching to get food from the trees, that information, that experience from the parent, was then directly passed on to the subsequent generation. Gregor Mendel was around basically during that time, and he had a different observation as an Austrian monk tending to pea plants, that kind of regardless of the different exposures, there were some traits that seemed to be passed down in pretty reliable mathematical patterns, suggesting that there were discrete units that were not necessarily directly influenced from the parent itself, that it was some different information that was being passed on. Now, actually, no, he was an Austrian monk, not necessarily in the nexus of the science of human genetics. So actually, during his lifetime, nobody really paid attention to his work. A contemporary of his, Charles Darwin, did form a theory on the variation that we see of traits and the exogenous pressures that might influence the traits themselves. But he himself was largely a Lamarckian. He had a kind of a parallel theory of just these general influences that would go on parents and then be passed down. And really, it was in the late 1800s, early 1900s that people kind of rediscovered Mendel's work. There's actually a lot of people that were trying to disprove what he observed who then said actually they were actually able to replicate his observations. One of those people was Thomas Morgan, who was looking at fruit flies, that he showed that when he mixed the fruit flies, that there were some traits that were seemingly unrelated that seemed to co-occur. And then he realized that these were probably traits or genes responsible for traits that were in close proximity to each other. And he was able to map out a linear chromosome for these. And these all were kind of related to discrete traits. Obviously, there are tons of continuous traits like height and everything that Ronald Fisher, who is credited for many aspects in biostatistics or statistics more broadly, gave us a theory that said that there may be actually multiple different genes for particular traits, and in an additive fashion may be responsible for some of the continuous traits that we observe. But really, kind of at this time, a lot of things that people were focusing on were, why people were tall, why some people weighed more, but also differences in IQ and differences in race. And there actually is a very complicated history that many of these initial observations formed substantial public opinion on race and were also led to pretty troubling policies also from the US as well. Now, over this time, people began to understand that there was a particular molecule that seemed to capture this information. And much of the emphasis has been, at least over this time, is trying to understand what the structure was to really understand the function. And this was a really interesting time in the 1950s because what had happened, World War II was done, and all the physicists that were working on the engineering aspects for World War II, many of them actually shifted their attention towards biological problems. And this is kind of the history for this discovery. There's kind of two groups. There's one in King's College, led by Lawrence Bragg. Sorry, that one was with John Randall, and then in University of Cambridge, Lawrence Bragg. Lawrence Bragg discovered basically X-ray diffraction with his father, got the Nobel Prize at 25, and still is the youngest for getting a Nobel Prize. And then there was John Randall, who developed basically radar and the root technology for microwaves. And similarly, there was Linus Pauling, who was very much a traditional chemist, who was working on protein structures and discovered alpha helices, beta-pleated sheets. And there was basically a race, because Linus Pauling at that time thought it was a triple helix for DNA. And meanwhile, because Linus Pauling was working on this and John Randall was disappointed that he beat them basically for protein structures, that he allowed James Watson and Francis Crick to start to think about DNA, whereas Lawrence Bragg very much was focused on using these technologies anyway for these molecules. And the reason why actually Linus Pauling thought that it was a triple helix, he's credited, or at least he has said, that is his images, the images that he had were actually not great images. And in the laboratory where kind of all the foundational technology was developed, Rosalind Franklin and her PhD student, Raymond Gosling, basically had the world's best images. And it's a very complicated history of how this image ultimately landed in the hands of Watson and Crick, but that led to the accurate description of the double helix. Anyway, I spent a lot of my time thinking about attribution and credit towards moving important projects forward, so it's not a theme that has gone away in science for sure. But anyway, this is a long-winded way of saying, so this paper is 1953, and Paul Dudley White's paper calling out the role for human genetics for medical conditions in 1957 is pretty remarkable. It's not like, this paper popped up on his Twitter feed or on TikTok or something, right? It requires some, deep thought for somebody to make that connection directly back to human genetics. And, before their time, there was recognition of really a canonical Mendelian disease, one that has a reliable inheritance pattern in familial hypercholesterolemia and its role for coronary artery disease, initially described as a dermatologic condition because its outward early manifestations are dermatologic, xanthomas primarily, and you see pictures here. And ultimately, it was a Norwegian physician who said that these patients also had high cholesterol. There was an increasing recognition of measuring cholesterol and its role in cardiovascular disease. And there was a co-occurrence of premature coronary artery disease for these patients. Now, in families in Lebanon, it was first described what the inheritance pattern was. This is incomplete dominance, which you may not have heard of, is autosomal recessive and autosomal dominant. It's called incomplete dominant because the heterozygotes have a severe phenotype and the homozygotes have an increasingly severe phenotype. And this was enabled because there's more consanguinity in certain parts of the world compared to others. So there are extended families that were described that had heterozygotes and homozygotes to describe this. Now, ultimately, the molecular defect was mapped to the LDLR gene, widely credited to Brown and Goldstein, who won the Nobel Prize, really focusing on familial hypercholesterolemia to understand the regulation of cholesterol. Now, Carl Mueller first described this as an inborn error of metabolism, and some were known at the time, PKU, galactosemia. These are all widely recognized, and pretty much all inborn errors of metabolism that relate to enzyme deficiencies are autosomal recessive, because if you lose one copy, there is some degree of redundancy. If you have 50% of the enzyme, you can pretty much stave away most of the severe phenotypes related to inborn errors of metabolism. But that was not the case. Both Goldstein was a physician. They're both physician scientists, right? So they already clued in, okay, maybe this is not exactly a traditional inborn error of metabolism, because heterozygotes were getting a phenotype. Homozygotes were getting a more severe phenotype. Now, this was not immediately clear how to go about studying this. There was already a knowledge that cholesterol regulation and synthesis was happening in the liver, but they couldn't easily go and just get like a bunch of liver biopsies. And already there was a well-developed model of using skin fibroblasts to look at inborn errors of metabolism, so just taking skin cells. So they applied those technologies. And they looked first at normal human skin cells, and then they gave they gave LDL particles, and they gave HDL particles, and they observed that only when they gave LDL particles, there was a suppression of HMG-CoA reductase. That was known as the rate-limiting step for cholesterol synthesis. And they took patients with homozygous FH. This is also what made it attractive for them to study, is that there were patients who were cleanly deficient in whatever this key regulating path was. Because there was already a knowledge before that there were examples that heterozygotes could have a dominant negative effect, where basically there is some change in structure and function for one copy of the protein that could affect the other copy. It was not necessarily just a clean defect, but the fact that there were homozygotes out there was helpful. So they got then fibroblasts for patients who had homozygous FH. And they observed that the HMG-CoA reductase levels were like 50-fold, 100-fold. They would give HDL cholesterol, obviously nothing would happen. They would give LDL cholesterol, and nothing really would happen. And in the normal cells, even just giving a little bit of LDL cholesterol did suppress cholesterol synthesis. That suggested some high degree of specificity of this regulation system. Now, one could say, okay, this is probably just a direct issue related to the enzyme, right? Because that is kind of our understanding of inborn errors of metabolism. But they did one extra step. They put cholesterol in ethanol that created this emulsion that could just go through the cellular barrier. And when they did that, when cholesterol could enter into the cell, that substantially suppressed HMG-CoA reductase levels to the normal. And that basically is what specifically clued them in to a specific receptor for LDL receptor. And there's obviously a bunch of additional steps, but it was kind of those like very simple observations, very clean experiments that led them to the LDL receptor. Now, the medicines that we use to treat high cholesterol or to lower cholesterol are focused on HMG-CoA reductase. These are kind of a separate discovery. This is Akira Endo, who's widely attributed for discovering statins. He was a research scientist at Sankyo, the precursor to Daiichi Sankyo, and basically discovered, like many different examples of fungi that had a specific defense mechanism against other parasites and other bacteria. And the compound ultimately was compactin or mevastatin. It was toxic to use in humans. The next generation, or at least the first commercially available one, was lovastatin, but its trade name was Mevacor, really, to give credit to this initial one. And the first patients to receive this medicine were those with familial hypercholesterolemia. And we know from the work of Brown and Goldstein and others that these medicines not only are inhibiting the synthesis of cholesterol, but because of the regulatory mechanism that they described, that also leads to an upregulation of the LDL receptors to improve the clearance of LDL particles from circulation. All right, so there's there's a fair amount of jujitsu to get from a genetic condition, or at least what we think is a genetic condition, without any genetic testing to identify what is the specific protein, and they ultimately cloned that gene. What changed was a massive effort and to ultimately understand what is the full human genome sequence, a necessary step to then go directly to a genetic test or a set of genetic tests to identify what the causal gene might be. In the US, there were at least 2 parallel efforts. There was a private effort and then a federally funded effort, which was actually budgeted for $3 billion, and that first draft ultimately cost $2.7 billion. In today's dollars, it's about $5.5 billion. Today, we can go to the Broad and others for $100 to get a whole genome sequence. And that's, it's been just about 24 years. So kind of the initial approaches then to take these kinds of families is an approach called genetic linkage, where we can take some of these markers because, obviously nobody has several billions of dollars to sequence, even 1 or 2 individuals. But we can take families and we can just sparsely select markers that are recurrently observed and see for people with disease, do they seem to get some of these markers, and those without disease, are they not getting markers? And ideally, there is one. Obviously, there's never 100% penetrance, but in a Mendelian family where there is severe phenotypes, and there could be, delayed onset of the phenotype, especially for adult onset conditions. But that had been the approach, and is to, to an extent, to some degree, still approach, but not so much these days. But that is kind of the route. But after you identify the marker that is segregating with the phenotype of interest, then you can go to the template and then just kind of sequence that area and see what you find. And that approach was taken to patients who had familial hypercholesterolemia, but did not have LDL receptor mutations. What was also known as another gene called ApoB, that's the primary protein of LDL particles, and missense mutations that disrupt the connection between ApoB and LDL receptor were known to cause a phenotype like familial hypercholesterolemia. But still, there were some that clearly had really autosomal dominant. There's no homozygotes, at least at the time, in these set of families. These were families in France. So they used the knowledge of the template for the human genome, and they started from the genome itself, and they basically used this technology of linkage analysis and positional cloning and prioritized a gene that nobody had any idea had any connection to cholesterol. That was PCSK9. Ultimately characterized as a gain-of-function mutation. It was ultimately characterized that PCSK9 is involved in the degradation of LDL receptors. So if you have more PCSK9, lower LDL receptors overall, and a phenotype that is similar to somebody who has deficiency of LDL receptors, a traditional familial hypercholesterolemia genotype. So, if these, gain-of-function mutations led to higher LDL cholesterol, the next natural thing would be, okay, what would loss-of-function mutations do? It would lead to lower LDL cholesterol. Lots of people were thinking about this, and it was also, like many things in science, there was healthy competition, a little bit of a race. People who ultimately won the race were Helen Hobbs and Jonathan Cohen, trainees of Brown and Goldstein, but obviously all stars in their own right. And because they knew something about PCSK9, they could do targeted sequencing of PCSK9. Helen and Jonathan were developing the Dallas Heart Study, they're still at the University of Texas in Southwestern. And they took the first 32 individuals that had the lowest LDL cholesterols, because still, pretty expensive to do. And they did targeted sequencing for PCSK9. They discovered that there were 2 loss-of-function mutations that were recurrently observed more commonly in individuals of West African ancestry. And they showed, then they basically specifically genotyped those mutations because that was more cost effective in the broader Dallas Heart Study, focusing on African Americans, and showed that compared to the average LDL cholesterol, these folks who had these loss-of-function mutations had substantially lower LDL cholesterol. Next natural question is, if they have lower LDL cholesterol, based on what we know about LDL cholesterol and cardiovascular disease, do they have lower risks for coronary artery disease? So they established a collaboration with the ERIC cohort. This is one of the NHLBI cohorts. Eric Boerwinkle at UT Houston and Jonathan Mosley, who's now at Vanderbilt. They worked together, and they just genotyped those 2 mutations in the broader ERIC cohort and showed that individuals had lower LDL cholesterol and substantially lower risk for coronary heart disease. And the timeline for this has been rapid. That paper is 2006. In 2016, we have demonstrably efficacious PCSK9 inhibitors that have been shown to do exactly what the human genetics suggest in a broad population: lower LDL cholesterol and lower risk for coronary artery disease. And there continues to be innovations in how to drug PCSK9. Now, going back to the observations from Dr. Levine, Dr. White, that there are numerous patients who have premature coronary artery disease. They have lots of them have family histories. Only a small fraction of them have phenotypes that suggest familial hypercholesterolemia. And some of the initial quantification of this comes from the Framingham Heart Study. Dr. White was among the people credited to bringing that initial NHLBI study to Framingham. And this was work led by Don Lloyd-Jones when he was a fellow at MGH, showing that independent of traditional risk factors, including like LDL cholesterol, just having a parent, a first-degree relative, led to about a twofold increased risk for cardiovascular disease, not explained by the things that we know today. Don has actually, as of this month, come back to Boston and is now the PI of the Framingham Heart Study. So to do this at scale, like, we can't obviously spend $3 billion on people. The innovation of doing a $100 genome is all relatively recent. But using that template and looking across broader populations, what are the markers that were currently observed, has allowed for the development of genome-wide arrays. That's basically the root technology for genome-wide association studies. These are, lots of colleagues that I still work with at the Broad. Stacy Gabriel is now the head of basically all the operations at the Broad Institute. Eric Lander is the founding director of the Broad and spent a brief time in the Biden administration. And then Mark Daly, who had first authored Nature papers when he was a high school student with Eric, remarkably brilliant and one of my K co-mentors. And then David Altshuler, Nate and my mentors, now the Chief Scientific Officer at Vertex. But these are obviously massive collaborations. I'm showing the bias towards at least the people I know who led these. But the Broad Institute has really been a place to generate these data and has been in a good position to really describe the data that they generate. But understanding what are the common recurrent markers where there is variation across populations has led to these arrays that are about like a million different genetic variants of the 3 billion variants that could be possible in the human genome. So in a genome-wide association study, you basically take individuals who you label as having the outcome of interest, coronary artery disease, and individuals who you label as not having coronary artery disease. And across those million genetic variants, you're just basically comparing the allele frequencies between the two. And then you can assign a test statistic. Test statistic is basically on the y-axis here. And now I'm sure many of you have seen these Manhattan plots. These are the first. And again, there's healthy competition, lots of people doing this, everybody converging on the same observation. And this lead signal has nothing to do with LDL cholesterol. Is in between some genes and has no relationship to any of the traditional cardiovascular disease risk factors, still remains the leading signal for coronary artery disease. Actually, a fair amount of work to unpack why this is associated with coronary artery disease, but it appears to be an important cause. And this is something that actually the field still continues to work on today. But because the data sets are getting larger, the methods are getting more efficient, we've now, as a field, identified a few hundreds of these gene regions. This is a circularized representation. This actually comes from the VA, the Million Veterans Program, which obviously has a very high burden of cardiometabolic disease, and a consortium basically just outside the VA. And you'll see that multiple different signals, most of these occur within genes because common genetic variation that influences outcomes appears to be within genes. There's a reason why we have our 20,000 genes, so it's not like there are tons of loss of function mutations and human knockouts that are out there. And these common genetic variants, each of these just modestly influence risk for an outcome. You see these effect estimates are within a pretty narrow range, right? Within about 10%. 10% is on the high side. But they do still provide an anchor for something that is broadly important because, as I said, these things are fixed features going forward. Now, HMG-CoA reductase also has a signal nearby, also has about a 5% increase. But as you can see, the effect estimate is one thing, but the fact that there is a signal still provides a very important basis for broadly relevant biology. And it turns out about almost 50% of these signals do also show association with traditional risk factors, but 50% don't. And that has been an important basis to really help us understand the residual risk for coronary artery disease. This is kind of an older representation of that, but as you can see, the extent of pleiotropy with the traditional risk factors and many that don't. Now, as I said, the method that many folks now have been using in trying to unpack that information for causal inference is kind of an approach that comes from econometrics, of using an instrument variable that is not directly related to the outcome itself, and that instrument variable can be kind of a proxy for the exposure. And here we use the genetic variance because you basically get, one copy of genes from mom, one copy of genes from dad, and it's pretty random which one that you get. And so you can use that randomness to basically kind of mimic, with caveats, a randomized control trial. And it turns out, people have been using this, now for probably about a decade. And then when mapping back to medicines that are approved, this is actually evidence from human genetics is a very important and strong predictor of successful approval of a medicine. Not everything requires human genetics, but it is very attractive for drug development. As I said, there are caveats because these genetic variants can also influence other things that may also influence the outcome itself too through pleiotropy, and there are many different tools to assess this, many different tools to account for this, but this is not necessarily a perfect test. One of the, probably is now really a classic paper, was really applying this, one of the early applications to lipids, and really a simple comparison between LDL cholesterol and HDL cholesterol because we all grew up in a world where HDL cholesterol was described as the good cholesterol, the LDL cholesterol as the bad cholesterol. Medicines, have now shown, at least that lowering LDL cholesterol lowers risk for cardiovascular disease, strong human genetic basis for that as well too. And at least some of the early parts, the inverse is true for HDL cholesterol. Individuals who have higher HDL cholesterol generally have lower cardiovascular disease event rates. So now we can use the instrument variable analysis to say, okay, what about the genetic variants that are directly linked to LDL cholesterol, the genetic variants that are directly linked to HDL cholesterol? Now, like in retrospect that we know from the clinical trials, genetic variants, common genetic variants that lead to increases in LDL cholesterol also lead to increases in coronary artery disease in proportional fashion. That's basically what this test is. And genetic variants that lead to increases in HDL cholesterol do not appear to change coronary artery disease. And this was published kind of in a very early era for HDL cholesterol therapeutics. And now, like billions of dollars later, we have now shown that so far that we've not been able to successfully pharmacologically modulate HDL cholesterol in a manner that lowers cardiovascular disease event rates. And that's pretty consistent now in retrospect with the human genetics. Positive human genetics for other biomarkers related to lipids have also been a strong spur towards drug development. One is lipoprotein(a). It's led by former medical student, very talented Rhodes Scholar who's maybe made great decisions, now venture capitalist. So here we see in a proportional manner, so lipoprotein(a) is an LDL-like particle, has been shown similarly like LDL cholesterol to be associated with future increased risk for cardiovascular disease. 1 in 5 individuals has this. Turns out uniquely that in general, that people who have high levels almost exclusively determined based on genetics. So it's pretty well powered for this kind of test. And similarly, genetic variants that are associated with LDL cholesterol proportionally increase risk for coronary artery disease. And now there are multiple phase 3 randomized controlled trials of medicines that have been developed that are very effective at lowering lipoprotein(a), and the human genetics predict that those clinical trials should be positive. Now, we can kind of do this increasingly on steroids. This was actually a very talented visiting Belgian student that was mentored by one of my former trainees, who's an assistant professor, who won the overall UK Biobank Early Career Award. That's like a global competition of hundreds of individuals. But this is one of the papers that he did looking at now increasingly downstream high throughput omics, basically the downstream products from the DNA, so transcriptomics, and then here is proteomics here, and connecting proteomics with a variety of different cardiovascular disease, and using Mendelian randomization, at least for those traits that are heritable, to say, okay, if we did clinical trials of medicines perturbing these pathways, these potentially novel pathways, would they reduce risk for each of these conditions? And some of these are conditions that actually have no demonstrably effective medicines like aortic stenosis. Now, there's obviously a rich history of pharmacogenomics already for cardiovascular disease. Probably the one that is most relevant to cardiovascular disease is CYP2C19, just based on what is known for the drug metabolism for P2Y12 inhibitors, patients who are often prescribed these medicines after they're coming out of the cath lab. And there have been subsequent innovations for P2Y12 inhibitors, really bypassing the metabolism that is required for clopidogrel to increase the efficacy and potentially the safety of these medicines. Now, meta-analysis of these trials have suggested the folks that benefit from those medicines, the newer generation P2Y12 inhibitors, are really the medicines that do appear to have deficiency in that pathway, loss of function mutations in CYP2C19, but those who do not seem to do just fine with clopidogrel. There doesn't seem to be a difference really related to bleeding. And in some scenarios, that can be considered. But that was really based on what is known about the metabolism of that medicine, but one can do these kind of unbiased screens to identify other genes that might be relevant, and this is kind of an example of that as well. Now, we still do lots of genome-wide association studies. It is still very challenging to unpack the mechanisms for this. Actually, Nate is leading a lot of the effort in trying to do this. But what is still easier is to find examples like PCSK9. That is really the poster child. It is much easier to understand, especially as it relates to mechanism and therapeutics. Now, over time, as I said, the costs continue to come down and the technologies have improved to do things like sequencing. And sequencing is necessary because for loss of function alleles, these are rare and they may be private to that individual or family or community, may not otherwise be predicted, and you can't just get away with 1 million markers out of a denominator of 3 billion. So you really need to enumerate each of these base pairs. Technology that is used is called next generation sequencing, which is different than the approach that was used for the Human Genome Project. And the field initially, again, also for cost, and because our hypothesis is that we really care about the 1% of the genome that is protein coding, has started with whole exome sequencing. And so our model is that if you find those rare variants that are associated with disease, because they have a direct impact on the transcript itself, on the direct function of the protein as opposed to levels more modestly, it is likely to have a greater phenotype. And there have now been very recently demonstrated examples of kind of new PCSK9s that have been developed by us and others, at least from the human genetics. So as I said, this is the example for PCSK9. Loss of function mutations really focused on targeted sequencing or targeted assessment of PCSK9 and effective therapeutics. And now other lipid-related targets really focused on triglyceride-rich lipoproteins, where individuals with loss of function mutations in ApoC3 lead to lower triglycerides. And you really have to sequence lots of different communities. You saw before, it's really serendipitous, and nobody knows why, that there are 2 recurrently observed loss of function mutations at an overall aggregate prevalence of 1% in African Americans for PCSK9. But in ApoC3, there's a founder mutation in ApoC3 that is more commonly observed in the Amish. About 5%. There's a nice science paper in 2008 showing that those individuals also have lower triglyceride-rich lipoproteins, lower burden of coronary atherosclerosis when they did non-contrast cardiac CTs. And then we showed through sequencing in the broader European population, less common, but we sequenced enough people to identify that there were people out there, they had lower triglycerides and lower risk for coronary artery disease. And then we found homozygotes. That was another paper as well, too. And now that there are 2 medicines, or actually there were 3 medicines that were effective, one that was never approved in the US because of toxicities related to thrombocytopenia, and now recently have approvals for familial chylomicronemia syndrome to reduce triglycerides and reduce the risk for pancreatitis. Actually, Nate wrote another nice editorial about that recently. And then ANGPTL3, also in a kind of a similar pathway, leads to lower LDL and lower triglycerides, and there are medicines that have now basically recapitulated that. The human genetics suggests that they should also lower risk for coronary artery disease. But because we sequence multiple different, actually the story for ANGPTL3, a lot of that starts in St. Louis based on an exceptional family that I think through a health fair discovered that they had an unusual pattern of lipids of combined hypolipidemia. Anyway, so we are sequencing lots of different populations, and through these studies we are finding that there are many other genes that in a very similar manner, that there are loss of function mutations that lead to lower amounts of lipids and in a manner that might also lead to lower risk for coronary artery disease. As an aside, because we have now lots of large biobanks that are phenotyped for lots of different things, as opposed to just the NHLBI cohorts that are focused on looking at cardiovascular disease, we can now start to anticipate potential on-target toxicities of these medicines as well. Also, as the data sets get larger, instead of kind of pooling the different, this kind of a methodologic requirement just to boost power, is that your power is tied to also what the prevalence of the exposure is, and as these things are pretty rare, so we kind of lump loss of function mutations together. But as the data sets get larger, we can assign test statistics to individual genetic variants, especially if we see them recurrently. And this is an example of such, and we can identify actually more genes through that approach. But this also does have direct clinical relevance because one of the clinics I have is a familial hypercholesterolemia genetics clinic. And for any genetic testing that is done today, you get this largely, feels kind of arbitrary, but certainly not quantitative designation of pathogenic, likely pathogenic, uncertain significance, likely benign, and benign. And that connotes lots of different things. Certainly when patients see pathogenic, for the most part, it's because they have a condition and they're saying that genetic variant is causing that condition. But increasingly, lots of people are getting testing for a wide variety of reasons. They're identifying pathogenic mutations who don't yet have a phenotype, but it almost feels very deterministic. It's very different, at least from the world that I'm in. And so as you suspect, many things that are classified as pathogenic have a large spectrum of quantitative association with LDL cholesterol and others. And we think that these data sets will really improve the counseling of these patients. And many things that are uncertain, we can actually now classify. So, that again, we talked about the mechanisms that we're trying to glean from the common genetic variants, still very challenging. We are doing increasingly rare variant genetic testing to find more PCSK9s, and that appears to be fruitful. Kind of another application has been to use these data for risk prediction, where we can kind of take the aggregate totality of information without really understanding the mechanism, but knowing that it is a predictor for cardiovascular disease to ultimately predict cardiovascular disease for a patient. Now, as I said, each of these common genetic variants only modestly associates with risk for cardiovascular disease, but if we put it together, it turns out it can be important predictors. And this is not unique to coronary artery disease. Genome-wide association studies are being done for lots of different cardiovascular traits, and we're involved in many of these. But what is an interesting phenomenon is that the genotypes themselves, the alleles that we have, are not changing, at least for an individual person. But our interpretation of the alleles are also evolving. This is not really that different from creating clinical risk prediction scores, because we had the Framingham heart study, the Framingham risk score, the pooled cohort equations, most recently the Prevent equations. They are all largely using the exact same covariates. But the weights are changing, how they're being integrated are also changing, and some of this is also secular differences in risk factor prevalences as well, and the importance of risk factors. But the scores are also generally getting better. That's really not that dissimilar from what's happening with human genetics, but our interpretation of the alleles is also refining. The methods that are being used are also to refine. Now, it turns out, we talked about FH as kind of the cornerstone for this. There are a lot of people who have FH. It's about 1 in 300 individuals, about a threefold risk. It's among the more common Mendelian diseases, similar prevalence as type 1 diabetes. But it turns out from a polygenic model, this genetic model, there are actually many, many more individuals that carry a similar cardiovascular disease risk that is not just tagged by LDL cholesterol that you'd be able to clinically identify. And this is kind of one metric of just the fold risk, because that rough prevalence for Mendelian disease has been threefold risk, about 0.5% of individuals. But basically with innovations in new scores, we can now find 1 in 5 individuals that has an FH-like risk. And that, so far, that metric is continually going up linearly, and I don't know yet when it will start to trail off. But it does require incorporating alleles that are seen in various different populations. That really is necessary to improve its prediction in all the populations. The US response to that is now the All of Us research program. We recently observed that still lots of Asian Americans are not participating. Asian Americans have a heterogeneous association with increased risk for cardiovascular disease, and we recently started our own biobank really focused on South Asians. Now, there have already been prescient studies, prospective studies of saying if you give these scores, these polygenic risk scores back to patients, what happens to patients and clinicians? This is a nice study that was done at the Mayo Clinic. where they took patients who were kind of intermediate risk based on these 10-year predictors, and then they basically communicated the clinical risk, and then they randomized some patients to also being communicated their genetic risk. And in general, you observe when people get risk communication, that leads to lower LDL cholesterol, but people who were told that when they and their clinician were told that they had high genetic risk, that led to lower LDL cholesterol. Actually, these investigators just posted a preprint a couple months ago. They've followed those patients now almost about 10 years, and those who were randomized to who had high genetic risk, who got this disclosure, had lower cardiovascular disease event rates. Now, we've done some assessments within the clinical trials themselves, because you could say, anybody you lower LDL cholesterol for is going to be beneficial, right? And that's true. If you look at subgroup analyses for folks who have diabetes, hypertension, high cholesterol, they all benefit from statins. They all have the same relative risk reduction, about 20 to 25% for cardiovascular disease event rates. But we do tend to prescribe more because those folks have higher clinical risk. Their absolute risk goes up. So the absolute risk reduction goes up as well too, right? Really driven just by that baseline risk. That's what's driving the number needed to treat smaller. Surprisingly, and not how they're designed, those who have high polygenic risk scores not only have higher absolute risk, but they have a greater relative risk reduction in cardiovascular disease risk with the same amount of LDL cholesterol lowering. And that further magnifies the absolute risk reduction, further shrinks the number needed to treat. So this is a pretty powerful tool, not only maybe compelling to patients as well, but in the end, we're doing these tests not just to do, just for a crystal ball. We're trying to do it to say, should we make a recommendation, for example, for pharmacotherapies? Now, this suggests that this may be a tool that may be particularly potent in saying that you may be more likely to benefit from this. And this seems to be a general insight with LDL cholesterol. Colleagues have done this now with the PCSK9 inhibitor trials. And in general, there is that same interaction as well, too. And we started to use this in the clinic. This is in the context of a research study. You can see on the x-axis here is polygenic risk score percentile. The people that we're ordering it for often are those who have a family history or are having premature events. They seem to have higher polygenic risk scores. And you can see some of the things that we think about has been helpful in initiating statins. Many of the folks that are coming to our program are also younger as well. Always will score low risk on a 10-year risk calculator that is basically all driven by age. The clinical risk factors kind of move the needle a little bit. But oftentimes we don't know. We do imaging and other things and surveillance. Now, there are other examples, really now fairly recent randomized controlled trials of just understanding, and NIH funded to some extent and VA funded, of understanding if you start to return this information to broader groups of individuals, polygenic risk scores, what might happen outside of these kinds of specialty clinics. And we've started to use this machinery to kind of develop in-house testing, which will come later this year. Now, I'm not going to do this justice, and I'm going to be very brief because I'm just wrapping up here, is that with the evolution of technologies and methods, as we've been doing sequencing in the research cohorts, which is largely based on blood, when you reanalyze that data, it turns out a lot of those folks also have acquired mutations beyond the germline mutations. And they have a prevalence much higher than anyone would have otherwise predicted. And many of them have acquired mutations that are cancer-driving mutations that have shown already increased fitness because they've expanded, because we were able to detect it with a pretty coarse resolution of exome sequencing and sometimes whole genome sequencing. It's about 1 in 10 individuals in their 70s. And these are strong predictors for blood cancer, but blood cancer rate is still pretty low in the population. And we and others have made some observations that it actually appears to have relevance to cardiovascular disease. But that has led to kind of a new area of genomics 2.0 of somatic mosaicism, which is obviously clear in its role for oncogenesis, but now acquired mutations that may have a role in non-oncogenic conditions. NIH has been very interested in this, so we're part of this consortium now to do deep DNA sequencing across multiple different tissues from autopsy specimens to understand the spectrum of somatic mosaicism across tissues in the human body. And we think that will be a new potential model for genetic disease, but it still is very early. Obviously, as we've begun to better interpret the genome, there's been in parallel development of technologies to manipulate the genome, at least for research purposes and increasingly for therapeutic purposes. Actually, the first CRISPR-based therapy was approved just about a year ago. It's for sickle cell disease. Great story of basically the story of genome-wide association study among patients with sickle cell disease, looking at severity of disease and separately levels of hemoglobin F. identifying an intronic variant at a gene called BCL11A, ultimately identified to be a repressive regulator of hemoglobin F. And that first one was a collaboration between CRISPR Therapeutics, Emmanuel Charpentier's company, and Vertex Pharmaceuticals, showed that in patients with sickle cell disease and transfusion-dependent beta thalassemia, if you basically CRISPR edit, this is outside the body, and then reintroduce those cells back in, those patients have greater hemoglobin F, are not getting hospitalized, or not getting transfusions. Actually, my wife was the lead for the beta thalassemia one, but that was led by David Altshuler. That's being applied to cardiovascular disease. It's first observations for TTR cardiac amyloid. And now coming full circle for PCSK9, if there are patients out there that are living without PCSK9 and appear to be doing just fine and are not getting heart disease, now, our former mentor is now the CEO of a company, Verve Therapeutics, and there are others actually doing this, of basically just permanently silencing PCSK9 through gene editing, through in vivo editing. Okay, so just to wrap up, we spend a lot on healthcare. This is not innovative insight. This is Dr. Califf, our recent or maybe still FDA commissioner, I'm not exactly sure, where he's giving grand rounds for us and showing this slide I've seen many times and maybe increasingly so now. And we really spend a lot of our time and effort basically on the end disease. Like I as a cardiologist, many of us as clinicians, we're taking care of patients often, if we're not in primary care, when they have developed disease themselves. And that is where drug development and science is really focused on. But really now we have the opportunity to take steps earlier and at least in new tools to identifying risk and mechanisms earlier. So now from Dr. White's observation in 1957, now with the American Heart Association's 100th year anniversary, lots of different observations, and then here by Dr. Braunwald, at least has called out now potentially a new opportunity, a new era for genomics and really in the prevention, the primordial prevention for cardiovascular disease. All right, so I know we don't have time, so I'm not going to read each of these things. We did cover a lot, and maybe there are questions, maybe there are not, but happy to take any at this time.
Thank you for that wonderful presentation. We have time for questions. Mark?
Great talk. Thanks for coming. When you think about risk, obviously doctors are doing that all the time. Patients sort of, my cholesterol's up, my blood pressure's up, it's down. But what's the storytelling that patients are doing internally with their genetic information, MIGs, long-term follow-up? I mean, that's very different than how most risk information, risk trials seems to bear out. I think, is it this internalization of like, this is me, I have to do something different?
Yeah, I mean, Mark, you probably deal with this as well, especially in primary.
Repeat the question.
Yeah, oh yeah. So the question is, I guess maybe if I'm understanding it correctly, is it like the concept of the genetic risk when the patients are getting it? Is there something different when there's a sense that it's innate as far as it requires modulation? Is that yeah? Right. Right, compared to at least a modifiable risk factor that you can potentially change, right? So I think people are very much clued in at least to the family history and the concept of genetics. There's already great, going on the other side, when people see that they have atherosclerosis, that can be a very powerful motivator to improve their risk factors if they have abnormal risk factors, or really think about prevention for cardiovascular disease events. It seems like genetics appears to do something similar, and I say appears because there haven't been enough studies to say this confidently. I think the MyGene study that I showed from the Mayo Clinic does suggest that it can be an important motivation. There's another study from Finland that suggests the same thing. People have tried to do this in diabetes, which is maybe kind of a few years behind. That has not actually been as compelling. They've applied this to pre-diabetics, and you may say pre-diabetics already have abnormal A1Cs and are necessarily on the path. But I think as far as risk communication, it can be an important tool, because the challenge in primary prevention, and the internists know this as well, if you make a recommendation, like if you prescribe a statin and the patient never has an event, it's either the medicine worked or it didn't work, and we'll never know, right? So people have to really understand the things that they're doing and why they're doing it, understanding the science. The human genetics, the fact that those features won't change could be a powerful reason why. I think my feeling is that it's a factor that is knowable very early in life, and we know that it's an important predictor for premature events. The 10-year risk scores don't pick up the premature events. So I think it is an important motivator early on, but we'll see.
Yes, sir.
I have a question and a comment. There is a difference between XX individuals and HDL in terms of cardiovascular disease, and XY individuals in terms of LDL. Those are very strong for a long period of time. That's my comment. My question is about your African data. Is there studies looking at the impact of dietary factors in terms of these different groups of people with the low LDL and comparing the dietary factors in those groups with low and high?
Yeah, so the sex differences, there are, so the question is describe that individuals who are XY and individuals who are XX, their relationship of their lipid distributions will be different, the associations with cardiovascular disease will be different, it's the comment, and then the...
We don't control for that in both of these studies.
Yes, I'll comment on that, because we've done some explorations. And then the second comment was, what about the diet, especially for those who have low LDL cholesterol, and maybe the risk for cardiovascular disease. So first, related to that comment, there are alleles that have been identified for coronary artery disease that seem to have a differential effect depending on sex chromosome, XX or XY. We don't know exactly why that's the case, but we have used those insights to also improve these polygenic risk scores, because it turns out, you know, the covariates, standard covariates are just adjusting for sex, but an overall polygenic risk score is actually better predictive in XY than in XX. Using the knowledge of the differential weights between the two different groups, which those data are increasingly being generated, does narrow the gap, but it doesn't narrow it fully. It suggests that we need to think about the methods to kind of bring this together. It is well recognized that these scores perform differentially across different genetic backgrounds, largely driven by the training data. It is, you know, very Eurocentric. But in general, most of these studies are relatively balanced between XX and XY, so it's certainly not being driven by that. So there's a lot more work to be done there. That's a great comment. And then diet, yeah, I mean, obviously diet has a substantial influence on the lipids themselves. There's been, you know, it's very hard to do these studies. You know, most of the studies that get published are based on epidemiology, and there are many different reasons why people eat certain diets and make the food choices. That often correlates with other lifestyle-related choices that they make. There have been small assessments of randomized controlled trials that are trying to look at, you know, very specific macronutrients in the diets and how that might lead to lower lipids. And based on the human genetics suggesting the causal role for things like LDL cholesterol, we would posit that other things, you know, non-pharmacologic things that would modulate the lipids should, in a very similar manner, if you're just controlling for that, also favorably influence risk for cardiovascular disease.
Okay, it's past the top of the hour, so I think we'll stop. Please, if you have questions, come down and talk to our speaker. Thank you again, Pradeep, for a wonderful talk.